Hey, what's up everyone? Hamza here. Today we're going over the lipids, which are essentially a family of nutrients. We've got the proteins, carbohydrates, and then we have the lipids. The lipids actually branch into two different types. First one being triglycerides and then the phospholipids. Now, both of these have got different structures, different functions, but they are both equally as important in our body. Cannot live without one or the other. Let's have a look at triglycerides first. You need to know the displayed formula. So it's just a formula of all the carbons and the hydrogens and the oxygens. So we're showing all the atoms and all the bonds. And what happens is we've got a glycerol molecule. That is your glycerol. And then we're going to have three fatty acid chains. That's the first one, second, and third. Each fatty acid chain has got a carboxyl group, so C-double-bond-O-OH from the carboxylic acids. So it's got a carboxyl group, and then it's got an R group, R1, R2, and R3. Now that is actually a variable group. It's basically a hydrocarbon, and it just means that it can be anything. The length can have an extra 10 carbons or just 3. It's variable. It can change. Now what's going to happen is we're actually going to have a condensation reaction. Condensation, we're condensing, we're putting two things together, we're joining things so that we have a chemical bond, and that's going to release water. So what's going to happen here is the OH from the carboxyl group and the H from the glycerol, those are going to leave. Now, because we've got three fatty acid chains, we're going to form three bonds, three ester bonds. The bond involved in lipids are ester bonds. Glycosidic is for carbohydrates, peptides are for proteins. Here we're dealing with ester bonds. And there's the ester bond. We've got C-double-bond-O and C-single-bond-O. That's the ester bond. Obviously we've got three of them, and we did remove three water molecules. Now, I've swapped R1 and R2 for just any random hydrocarbon. And you can see that the difference here is that this one does not have a double bond between the carbons. Here there is a carbon-to-carbon double bond. And it's a bit slanted as well. I'll explain that in just a bit. So this is actually a saturated fatty acid chain. A fatty acid chain that only has single bonds. This is an unsaturated fatty acid chain. It has double bonds as well. And the double bond is actually going to cause a kink, just the carbon to bend a little bit, and these types of fats are found in different places. You might have seen it. Saturated fats are found in butter, cream, whereas unsaturated fats, we're talking about fish oil and any plant oils, avocados. So these types of fats are found in different places. Now that was the displayed formula. They may not always ask you to draw this entire thing with a glycerol and all the fatty acid chains. Sometimes they might ask you to just draw it in simple terms, and that's what a triglyceride looks like. So what do we have here? We've got our glycerol, this little oval shape. We've got our three rectangles which represent the chains, and we've got these three lines which represent the ester bonds. Now, you'll get asked this a lot. Lipids, triglycerides, what are they used for? The triglycerides are actually used for three different things. Number one, they are used for energy storing. They are used as energy storing molecules. They can release energy by breaking the ester bond. So if you want to get energy out of this triglyceride, you just have to add water molecule. You've got to add a water molecule to break a bond down. This is called hydrolysis. So hydro, water, and lysis to break it down. Hydrolysis is the breaking down of a chemical bond, the ester bond, and that will actually release energy. So that will separate the glycerol and the fatty acid, and this bond's going to be broken. It's going to release energy. So number one, it is used for energy storing. Number two is a really, really important one. It's also used for insulation. So what happens is you've got this layer underneath your skin. We can call it subcutaneous fat. It's a layer under the skin that provides you warmth. It provides you insulation just to keep you warm. And the final thing, which is really, really important, is for protection. So what happens is the triglycerides Altogether, they form a tissue called adipose tissue. So they work together and they actually wrap around your vital organs. We talk about the heart, the liver, the kidneys. And they do so to give protection. They absorb any shocks. So if you fall or someone just pushes you, your heart's not going to shake. It will actually stay in place because of this. It's called adipose tissue. So it is a layer that wraps around the heart to absorb any shocks. And it's very thin. However, for someone who's got a very poor diet, they have too much junk food, oily and fats, then the tissue will actually be very thick. And that can actually be dangerous because that will put a strain on the heart, that will put pressure on the heart. Right, let's move on to phospholipids, which is the other type of lipids. Now the phospholipids, phospho meaning phosphate. What just happened here? Well, it looks pretty similar to the triglycerides, but it's only got 2 triangles instead of 3. Yes, so phospholipids only have 2 fatty acids. And instead of having a third ester bond with another fatty acid, it's going to have a bond with a phosphate group, which is why we call it phospholipid. And actually what they look like is this. Think of that being the glycerol, and these are your chains. Now, you might already know, phospholipids are used to form the bilayer of the cells. So you've got the cell, and we've always been told that there's a cell membrane. Now, the cell membrane, it lets stuff in and out. Well, specifically, it's these guys that let stuff in and out of the cell because they basically just form around the entire cell. And there is reason behind it. You need to know about this. Now, when you draw your diagram, because you will be asked to draw a diagram, that would be wrong. That would be wrong. Make sure that each fatty acid actually connects to the glycerol. And what happens is, we call it a bilayer because on both ends, we've got phospholipids that are facing in opposite directions. What you need to know is that this is the head, the glycerol, this is the head of the phospholipid, and these are the tails, the chains, fatty acid chains. You need to know that the tails are hydrophobic. Hydrophobic, from the word phobia, fear. They're actually scared of water, so they do not want to come in contact with water. Whereas the phospholipid heads, they are hydrophilic, water-loving. Now, in the exam, don't say they are loving water. Do say that they are attracted to water. And so what's going to happen is they will push inwards. The tails are going to face inwards because they are scared of the water that's just on the outside. And the heads, they are facing water because they are hydrophilic. That is the use of a phospholipid. Now, what you also need to remember is that sometimes we can have cholesterol joining the party. Cholesterol might bind to the tail. Cholesterol binds to the tail of a phospholipid. And cholesterol is actually a good thing, unless you've got a high concentration of it. Cholesterol, what it makes sure is that this phospholipid bilayer is smooth. There's a nice flow to it. It's making sure that everyone is kind of close to each other, not too close and not too far away from each other, just so that there is a nice flow and we can let stuff in and out. If the phospholipids are too close together, it's not going to let anything in. And if the phospholipids are too spread out, it's going to let everything into the cell. So we don't want that to happen. Cholesterol basically regulates the fluidity. It regulates the fluidity. Now, just a bit more about the phospholipids. They form this bilayer, and the bilayer is said to be fluid mosaic. And it's a 2-mark question in the exam. Why is it said to be fluid mosaic? Well, fluid because there's a flow to it. Okay? There's a flow to it. They're not just stuck in one place like a solid. You know, particles in a solid are just stuck stationary. Here, they move around. They move around. And why is it said to be mosaic? Mosaic, just like a jigsaw, a puzzle. We've got little pieces of everything. We've got a bit of phospholipids here. We've got the cholesterol that we've mentioned there. We have this entire thing. I'm not sure what that is. That is a transport protein. I'll talk about that in just a bit. And we have some glycolipids, glycoprotein. So it's like a puzzle. There's not just the phospholipids, there's a bit of everything here. And everything, they all work together, all these molecules work together to let stuff in and out. So first things first is, it depends on what substance is going to go into the cell. So for example, we've got oxygen. Oxygen is small enough, it is a small, non-polar molecule. So oxygen can actually, if it wants to go into the cell during gas exchange, it can just pass through the membrane. The phospholipids are going to let it through. Yep, you can go through because you're small enough and you are non-polar. You don't have a charge, you're not an ion. You're not a positive or a negative ion. You don't have any charge, we'll let you through. However, what if glucose wanted to join? Glucose is such a big molecule. Phospholipids are going to say, hey, sorry, yes, you don't have a charge, that is fine, but you're too big. So you're going to have to go through the transport, please. And it's going to pass through the transport protein. What if we have sodium? Na+. Phospholipids are going to say, you're small enough, but you've got a charge. We can't let you through, I'm afraid. And so it's going to pass through a transport protein. So if you want to pass through a phospholipid, you must be non-polar and you must be small enough. And you must also be lipid soluble. So you must be able to dissolve in the phospholipids as well. So for example, estrogen is coming, or any steroid, or fatty acid would be the easiest example here. The fatty acids could pass through here. Any substance that is not lipid soluble, it can't pass through here. Final thing, the glycolipid and the glycoprotein, you rarely get tested on this, but just for your information, they act as a recognition site. So a glycolipid is basically a sugar with a protein or a lipid with a protein. And what they do is they act as recognition sites. They send signals and they retrieve signals from other cells. And that's how one cell communicates to another. Final thing is the emulsion test. How do we test for a lipid? Well, you must add your unknown in a grease-free test tube. Make sure that you don't get your fingers on the inside of the test tube, because our skin actually produces oils, and that could give us a false positive. So make sure you pour it in a grease-free test tube. And then you're going to add ethanol, shake it once. Then you're going to add water, shake it again. And the final thing is, you should see a cloudy white layer that forms, and that would give you a positive result. The unknown did have some lipids, and that is it. If you've got any questions, drop it in the comments. Make sure to subscribe because you don't want to miss out on the entire spec, and I'll catch you in the next one.